Welcome. My name is Nat Hovey and I'm your host today. In April last year, we launched Alfa Laval CultureOne to the single-use biopharma industry. And now, more than a year later and multiple trials at multiple customer sites, we want to share the findings and how we are delivering value to the customer. And with me in studio to share these findings, I have three of my dear colleagues. It's Doug Osman, who is our Director for Life Sciences here at Alfa Laval, has worked many years for Alfa Laval and has been part of this development from the start. Michaela Sievert, who is our Global Launch Manager. She will share our white paper findings that we did together with a customer. And thirdly, and not lastly, Petter Torvid, who is a Concept Development Manager in our R&D department and has of course been part of this development from the start. So Doug, we start with you. In case we have people joining this webinar who have not listened to the previous ones, would you mind making a short recap of what we launched in April last year and what our starting point was?
Yes, in April of 2020, we launched the CultureOne, the first premium single-use separator for the harvesting of biotherapeutics. We saw a need in this category as an extension of our existing stainless steel product line in order to deal with the evolved materials that our customers have been producing. A lot of upstream advances have resulted in higher drug concentrations, higher cell concentrations, and the existing solutions for harvesting this material were really being pushed to the limit. So we responded then to a market demand for something that would deal with these changed and evolved materials, and also looked into a little further down into the future to make sure that a solution we would generate would be applicable for even the next generation of changes that we see our customers pursuing. So that in summary is the motivation behind producing the CultureOne, both from being able to fit in a single-use environment, but also to deal with the changes to the materials from the upstream processes.
So in developing an improved solution against those process challenges, what are the most important criteria?
Well, first and foremost for our customers, it's product yield. Any recovery method must deliver a high product yield in this first step, because losses in this first step have a multiplicative effect in terms of the overall yield from the efforts that our customers make to produce these processes, or these materials, I should say. And then we're looking at how the quality of the materials that we produce from this first step affects the downstream operations, various filter and purification operations downstream. So we wanted something that delivered a high quality material after the separation result. And then, finally, we were looking at something that would treat the material quite gently, so that we would not introduce additional impurities that would have to be dealt with downstream as well. So these three criteria, yield, downstream filterability, and low additional lyses, were the primary considerations in terms of delivering a good result out of this particular process step.
Any additional consideration we brought into the development?
Well, we wanted something that would operate on a continuous basis, so that we could reach a steady state and have something run in a predictable and reliable way. We also wanted something that would deliver this process result without any additional additives, because additives, they complicate operations, and they add expense, and they add time, and they add risk. So we wanted something that would operate without the need for any chemical addition. And we also wanted something that would be quite simple to operate as well, because a lot of these operations involve portable equipment, and having something that would operate quite easily and simply in that type of environment is very important to make sure that the results that are achieved are repeatable every time. So those are some of the additional considerations that we looked at in terms of having something that would function well in this type of an environment.
So after a year working closely with the customers, what are the feedback that you hear, Doug?
Some of the things that we're hearing back are exactly what we had hoped to hear, that we do have something that delivers a fantastic process result that is simple and intuitive to work with, that produces repeatable and reliable results, and really delivers on the things which customers value in this particular process step. So we've heard other comments such as it treats the material quite gently. It's a very quiet system. Doesn't generate a lot of noise and is easy to work, right next to. It's portable and straightforward to work with, and really kind of intuitive, and provides fantastic feedback in terms of what exactly is going on while you're making a process run. So we've had a lot of good comments back, and it's really been great to hear that, because it has validated that we have delivered against the original design criteria.
Thank you, Doug. So what I hear you say is that we took departure in the problems that our customers are facing when we created the solution, and the feedback that we get is very, very positive. That's good to hear. Thank you. We also have documented results of the performance that we can deliver. We published a white paper late last year together with the customer. Michaela, welcome. Would you mind telling us a little bit about this initiative?
During the development of CultureOne, we had a very strong partner in Genentech, one of the leading biopharmaceutical companies based in California, US. And together we ran numbers of tests, a lot of trials. We did that on both commercial cell culture material and also their products under development and under real world conditions. And during the tests, we looked closely at process key factors like yield, like filterability, additional lyses, and the results turned out to be very good. And together with Genentech, we published these results. We made a white paper, and that was recently published by Bioprocess International.
You wanted to highlight 3 things from the white paper. You talk about product yield, low additional lyses, filterability, which is also what Doug mentioned earlier and said is important to the customer. If we take it one by one and start with product yield, what does this mean, and what results did we see?
The product yield is the percentage of valuable protein, so this is a real key factor. And for all the tests that we did, under all conditions, we were above our target of 90% product yield. And we have also run further tests and reached up to 98%. So that result is very good. The secret behind the reason for this is basically the centrifugal technology is a very efficient separation technology. And with the right rotational speed, plus the high separation area with the latest disc stack design, there is the reason behind this result.
Secondly, we wanted to highlight low additional lyses. Could you tell a little bit about what that means and also the results that we saw?
Additional lyses is the amount of cells that get damaged through a process step. And in our case with CultureOne, we saw that lyse cells were typically very low, well below the target. Even at higher flow rates, we saw that the additional lyses level were as low as 5%. And these results, again, is typically connected to the optimal rotation speed and also the minimized shear on the fluid, which you get with the top stream design.
And finally, we wanted to highlight filterability. Again, can you tell what it means and also what results that we have documented?
Filterability or filtration throughput is the filtering characteristic of the fluid after centrifugation. And this is essential for the downstream processing, what happens after the separation step. And that includes sterile filtration. So our tests showed that we can achieve as high as 900 liters per square meters in filterability. And those results were actually comparable or better than what we see for any baseline process. And the key, again, is a very efficient centrifugal separation or removal of cells and debris, which is connected to the hermetic design and the top stream.
What you mentioned now is the results published in the white paper that we did together with Genentech. Would you say that these results are standalone, or have we been able to repeat them?
Well, we have had continuous proof of concept for CultureOne. We have continued to document these level of performance, and we have repeated them with more than a dozen of customers by now. So in my mind, the R&D team, together with Genentech, during this concept development, has taken us a great step further. And now CultureOne is a unique fine-tuned design concept based on a lot of test results, proving that hermetic design, top stream, and Unidisc can really contribute to a consistent and efficient cell harvesting process.
So thank you, Michaela. And the white paper shows we have very high product yield. I heard you mention as high as 98%, which is really, really good. And also that the customer product has very high quality after the separation. And for me, as a non-biopharma expert, it sounds that the customers get high quality product and more of it in this separation process. And that's good. Thank you. Welcome, Peter. You are our concept development manager at our R&D department, and you have been part of this development from the start. Can you share a little bit about the approach that we have had to product development in this process?
Well, it's been an agile approach together with the customer. We have already from the first meeting and the first field test, we modified our test centrifuge, the Explorer. We made it into a somewhat of a single-use device. We went to the customer and we tried some of the new features, blending with some of the knowledge that we have since the past. And since the cells that we are separating is not readily available, there is no test media. We had to understand how it actually worked in real life. And early prototyping and step-by-step working with the customer towards the final product was the approach and something we succeeded with.
So Peter, why could we do this?
Well, it's because we didn't everything new. We didn't do everything new. It just looks different, but it's still the same separator principles that Alfa Laval has used for centuries. Hermetic technology, together with better disk performance with the Unidisc, and also a new development called Topstream, where we treat the cells heavy phase in a different way. That's development we started a few years back, and now when this possibility came up and this need from the customer, we blended the technologies together and could approach this development task in quite a speedy way and to the point. So it's blending new with old in a new package.
So what would you say is the next step now?
Well, it's to continue on the path that we have set out, continue to work with the customers, understanding what's around the corner, what the future needs are, and at the same time showing them what is possible to do. Now, when we have a collaboration, we know that prototyping works in their industry. We can then show new values and other ways of using our technology to solve their future problems. So in the collaboration with the customer, a lot of exciting things can happen when we work together, and we really are looking forward to the future. We're here to stay.
Thank you, Peter. So in summary, we have taken departure in separation technology that we know well, and we have blended in unique Alfa Laval technologies that we know deliver results, and we have created something completely new. This is great. Doug, I have one final question for you. Can a customer now use this product in their GMP production environment?
Yes, one of the most important criteria for customers is to have process context surfaces which can be validated against the appropriate, regulatory authorities in different markets around the world. And we've done all this work, all the laboratory studies, all of the sterility studies against it, and we're happy to say that the CultureOne is very well suited for operation in a GMP production environment.
So thank you, Doug, Michaela, and Peter for joining us today, and thank you for listening in to this webinar. And if you would like to know more about our product and what we can do for you, please reach out to your local Alfa Laval office in the country where you are, or you can check this page out and fill in the form, and we will guide you to the right person. So thank you again, and stay safe and healthy.